a very vast one, can be only touched upon. Each one of the sections of this discussion could occupy a week's symposium. But nevertheless, what we hope to accomplish is to provide a broad framework and to have selected experts describe how certain areas in this large field are being probed. One only has to contemplate upon the significance of the immune response in protecting against bacterial and viral infections to appreciate the paramount significance of this system in health and disease. Impairment of the immune response, which accompanies certain disorders of development, or which follows the ravages of certain diseases, or the treatment of certain diseases, is a subject which is attracting much attention and which could alone occupy our time. To go further there, however, there is much evidence that the immune system is a two-edged sword, and that under certain circumstances it can be turned against the body, giving rise to diseases with autoimmune factors at play. It should be emphasized that because autoimmune factors can be detected, this does not give them any primacy in the causation of a specific disease. Nevertheless, current thinking imparts autoimmune phenomena a role in the cyclical progression of certain disorders. We can consider such diseases as rheumatic fever and acute glomerulonephritis of children, streptococcal related, in which the streptococcus is the inciting pathogen, but in response or in reaction to this infection, a cyclical autoimmune process is set up, giving rise to many of the manifestations of the disease. Hashimoto's thyroiditis, rheumatoid arthritis, lupus erythematosus, myasthenia gravis are examples of diseases where such mechanisms are believed to play an important role, although they may not be etiologic in any sense, and the inciting or triggering pathogen or mechanism is entirely unknown. In the hematological area, there are certain groups of disorders in which mechanisms of this type prevail. This first slide briefly summarizes a few of these. In certain instances, such as drug-induced thrombocytopenic purpura, the mechanism is quite clear. An interaction between drug and platelet occurs with a hapten-like complex which renders the platelet antigenic and incites or elicits antibody production, which in turn destroys the haptenized platelet. In other instances of thrombocytopenic purpura, such as following streptococcal or viral diseases in children, such a mechanism can be inferred, and there is some evidence that the platelet is coated with antibody. However, the exact relationship between this type of process and this remains obscure. In terms of hemolysis or hemolytic disease, we frequently see autoimmune hemolytic anemia in association with viral or pleural pneumonia-like organism infection, in association with lymphomas, lupus, rheumatoid arthritis, and in many of these instances we can demonstrate coating globulins on the red cell, and by analogy to a known hemolytic disease of this type, Rh incompatibility leading to hemolytic disease of the newborn, we can infer that the immune system is leading to autoimmune destruction of the red cell. However, this is merely an inference and is not proven. In other situations, the relationship between drugs and other pathogens and aplastic anemia seems to be becoming clearer, although much remains to be done in this area. In any event, because of the scope of the immune system in the relationship to the development of various diseases, it behooves us to scrutinize the system as closely as follows. We might begin with asking the question, what is an immune response? And on the next slide, I've listed a few of its characteristics. In the primary response to an injected antigen, there is a latent period during which there is clearing and processing of antigen in some manner, dependent in part at least upon histiocytic elements of the reticuloendothelial system, and possibly in some other instances upon the polymorphonucleoleucocyte. There is proliferation of peripheral lymphocytes in lymph nodes and the appearance of plasmacytic or plasmacytoid cells. These cells have a characteristic heavy basophilia and stain heavily with pyronin stains, are often referred to as pyrenophilic cells. The basophilia and pyrenophilia is related to RNA synthesis and protein synthesis in these cells. There is enlargement of germinal centers of lymphoid follicles and the appearance of new follicles, especially prominent in areas of lymph nodes regional to the antigen injection. There is increased RNA turnover and protein synthesis in specific cells, as mentioned, with the early appearance of a heavy molecular weight 19S specific antibody, followed by the appearance of 7S specific antibody. The immune system is characterized by immunological memory, whereby a second exposure to antigen elicits an accelerated and augmented antibody titer, the anamnestic response. Delayed hypersensitivity shares many of the features of primary type of immune response. The proliferative response in the reticuloendothelial system is not unlike that in the primary reaction. However, circulating antibody is of questionable significance, and more characteristic of the reaction is a local mobilization of lymphoid cells into the area of the hypersensitivity response and proliferation of cells in this area. Passive transfer of delayed hypersensitivity requires cells, or at least cell constituents. This phenomenon is also manifested by immunological memory. The next slide is from a paper by Ur and his associates and summarizes the biphasic nature of the serologic response to an injection of antigen, in this case a bacteriophage. There is clearing of the antigen, fixation in the reticuloendothelial system during the latent period, followed by a rapid rise in heavy molecular weight 19S globulin of transient nature, and this is replaced by a rise in 7S antibody, which persists for variable but generally prolonged periods and is characterized by an augmented and accelerated rise in titer on second exposure to the antigen. Clearly, the lymphatic system is a key element in this response. It is certainly not independently operative. It probably does depend upon a coordination with various other parts of the reticuloendothelial system. Some of the features of the peripheral lymphatic system that relate to its role in immunity are described in this next slide. The lymphatic system is far-flung, as we all know. It is self-sustaining in that all peripheral lymphocytes, large or small, appear to be capable of proliferative response under appropriate conditions. It has access to most areas of the body. The lymphocytes are characteristically recirculating cells, which may be formed in organized area of the lymphatic system, enter the lymphatic flow and bloodstream, pass through tissues, and return to lymphatic organized structures. The lymphocyte has the ability to persist or exist for long periods in an inactive state, and this is thought to relate to the phenomenon of immunological memory. And lastly, it has this characteristic proliferative response to antigen processed in some appropriate fashion. It is becoming quite clear, however, that all small mononuclear cells that look like lymphocytes may not show this potential. There is an old Chinese proverb which says that you shouldn't select your donut by the size of the hole. We recognize a lymphocyte not because of what it is, but because of what it isn't. It lacks any of the distinguishing, differentiated characteristics of other cells while it is in the state of a resting small lymphocyte. There is good evidence that small lymphocytes in the thymus and bone marrow, for example, are less reactive than lymph node or spleen cells. An example of this is shown in the next slide, which is from data of Billingham and his associates, in which the persistence of injected thymus and bone marrow lymphocytes in newborn recipients and the ability of these cells to produce tolerance in these animals is far less than cells from lymph nodes. There is additional evidence to show that cells in the thymus and bone marrow have differences in a kinetic sense from those in the circulating blood and peripheral lymph nodes. The question of the role of the thymus in immunity could comprise another seminar. Suffice it to say that the possibilities include at least the following. Next slide, please. There is considerable evidence that there is a hormone secreted by the fixed epithelial cells of the thymus, and that this hormone is necessary for the proliferative this hormone secreted by the fixed epithelial cells is necessary in order for the peripheral lymphatic tissue to undergo the proliferative response to antigen that I have mentioned earlier. Another possible role of the thymus is as a source of lymphatic cells which can become directed in an immunological sense upon reaching the periphery, but which are inert while in the thymus. There is other suggestion that the thymus may be an area of selective removal of unwanted clones of cells. There is much to make one think that in the adult state, the thymus is merely an effete organ which represents a vestige of primitive hematopoiesis. These are the various considerations as far as thymus participation in the immune system is concerned. To summarize these opening remarks, we can outline the immune process briefly in a manner such as that shown in the next slide, in which an antigen is cleared and processed. There can be a derangement at this level, either in terms of recognition of the antigen or alteration of the host tissues to become antigenic. Antigen, in general terms, leads to a proliferative response. There may be an impairment of the proliferative response, spontaneous or as a result of illness, leading to immunological deficit. Differentiation of the cells resulting from this proliferative response may be impaired in the direction of defective immunoglobulin synthesis or in the direction of impairment of the cell-mediated cytotoxicity characteristic of the delayed hypersensitivity reaction. We will present a case, may I have the lights please, in which a common disorder is associated with a severe derangement of the immune system. Dr. Winkelstein will now present this case briefly.
May I have the first slide, please? Mrs. Ann Gellis, 53-year-old white widow, who has been followed at UCLA since June 1964 for chronic lymphatic leukemia. Her history is outlined on the slide. She was first discovered to have this disease in 1956 when she sought medical assistance for menometrorrhagia. She received no therapy for her leukemia until 1960, but did receive a course of pelvic irradiation. In 1960 and again in 1962, she had episodes of severe acute hemolytic anemia. With the first episode, her hemoglobin dropped to 5 grams %, and she required 15 units of blood. The second episode was milder in that she did not require transfusion. Both episodes were treated with steroids in moderately high doses, chlorambucil, and splenic irradiation. Following her second episode, maintenance therapy with steroids and chlorambucil was initiated. She moved to Las Vegas in 1964 and came to UCLA for further follow-up. Physical examination revealed a slightly obese white female who did not appear either acutely or chronically ill. Positive findings included scattered inspiratory and expiratory rhonchi in both lung fields. The spleen tip was palpable 12 centimeters below the left costal margin, and the liver 5 centimeters below the right costal margin. There was generalized lymphadenopathy. The remainder of the examination was unremarkable. May I have the next slide, please? The laboratory data is listed on this slide, as positive findings are marked with an asterisk. Of particular significance is that she has maintained a normal hemoglobin throughout her follow-up period. The white count has ranged from 40 to 140,000, with the differential count showing a marked predominance of small and medium-sized lymphocytes. Platelet count and reticulocyte counts have been normal. Other significant findings include a positive direct Coombs test, which later reverted to negative, a positive test for cryoglobulins, and a significant depression of the gamma globulin fraction of the serum proteins. Uric acid was slightly elevated to 6.83 milligrams percent. Bone marrow aspiration showed 86% lymphocytes, consistent with the diagnosis of chronic lymphatic leukemia. Since she has been followed at UCLA, her course has been characterized by repeated episodes of purulent bronchitis, following even mild upper respiratory tract infections. Except for this, she has had no further difficulty with her leukemia. Her current therapy includes chlorambucil and every other day steroids, plus intermittent antibiotics for recurrences of infection. The lights, please.
This case of chronic lymphocytic leukemia is a common form of leukemia in approximately 15% of patients. We encounter deranged autoimmune phenomena characterized in this instance by hemolytic anemia, Coombs positive in type, and also in this instance, despite the fact that this woman has a plethora of small lymphocytes, which are, we have proposed, a key cell in the immune system, has a gross defect in immunoglobulins and marked susceptibility to common bacterial infections. I think we should, at this juncture, go into, in a little more depth, some of the evidence for thinking that the small lymphocyte is a key cell in the immune mechanism. And Dr. Hildeman, who has performed a good deal of work in this area, will present some of this evidence now.
Over the years, lymphocytes in general and small lymphocytes in particular have been thought to perform almost every cellular function known in the body, and at various times have been considered either multipotential stem cells or even effete end cells. We now know that lymphocytes have considerable proliferative activity. There's much, however, about the life history and functions of lymphocytes that still remains to be defined and established. I want to consider briefly with you some of the recent and long-standing evidence concerning lymphocyte involvement in immune responses. As many of you are no doubt aware, studies over the last few years on the phylogeny of immune responsiveness show that vertebrates in general, down through the fishes to the level of cyclostomes, are immunologically competent in the fullest sense of the word, and this potentiality is associated with three main components. First of all, the presence of a thymus gland, the existence of a family of lymphocytes, and finally, the capacity to produce immunoglobulins, specific immunoglobulins in the face of diverse antigenic stimuli. The development of lymphocytes has long been studied, and the picture of their appearance during ontogeny we studied in some lower forms, and in general the picture is quite similar to what others have found in higher animals, including mammals. First slide, please. Certain species, such as the bullfrog and amphibian, which have prolonged periods of embryonic or larval development make very suitable material for studying the developmental appearance of cell types. And the picture we found here turns out to be pretty much characteristic of what has been observed at various times in birds and mammals as well. Erythrocytic cells appear fairly early in the game in association with monocytes and large and medium lymphocytes. The last cell types to appear in development are the granulocytic cells and more especially small lymphocytes. We find that the capacity of immunologic self-recognition, particularly in terms of the response to homografts or allografts, appears developmentally at about the stage of development when small lymphocytes put in their appearance. So this kind of circumstantial evidence for involvement of small lymphocytes in immunologic responsiveness has been known for a long time. Next slide, please. Now, the more direct evidence of the role of lymphocytes comes from the work of Gowans with thoracic duct lymphocytes in rats and our own work with peripheral purified small lymphocytes in mice. We've independently established that highly purified small lymphocytes taken from adult mice on injection into newborns of another strain differing for major histocompatibility antigens will lead to the development of transplantation disease. That is to say, these small lymphocytes alone are capable of mounting an immunologic attack on the immunologically immature newborn mouse, and one commonly finds that the animals become ill and proceed to die at about 8 or 9 days of age. The illness of transplantation disease is a very characteristic one associated with runting and diarrhea and many internal pathologic changes, which I'll illustrate in a moment. One gets the same picture here of multiple deaths in acute transplantation disease during the second and third week of life in various mammalian species and with mixed populations of lymphocytes as well as peripheral leukocytes. It's of considerable interest, though, that small lymphocytes alone are capable of producing all of these reactions. You'll note here that essentially all of the animals being attacked in this way are killed within 30 days. An occasional animal will survive longer. Next slide, please. This illustrates an occasional long surviving chronic runt. This is an experimental newborn A-line mouse that was injected with adult C57 black small lymphocytes on the day of birth. This is a litter mate control. This was taken at 48 days of age, and you note the very dramatic difference in the condition of these animals. In these strong strain combinations, the result of the lymphocyte inoculation is almost invariably death of the recipient animal. Next slide, please. Now, we were intensely curious about the very early changes in transplantation disease that might account for the mortality observed. And we were particularly perplexed because the effect of the injected cells seemed to be very early, whereas the deaths did not appear generally until a second week in life. So a year or so ago, Drs. Walford, Gallagher, and I proceeded to investigate the early histopathologic changes associated with runt disease. For this purpose, we used the adult lymphoid cell into a neonatal recipient model and evaluated the changes in lymphoid tissues from soon after injection up until the time when the animals were actually dying with the disease. Here you see plotted on the ordinate the so-called indices of graft versus host reaction, which represent the relative body weight or organ weight of injected experimental animals over the respective weights of the of litter mate controls. The hatched area represents the normal range around a value of 1, which would indicate no significant change. You see here that both with mixed spleen cells and with highly purified peripheral small lymphocytes, that within 48 hours after injection there is a depression of thymic weight generally associated with an enlargement of peripheral lymphoid organs, particularly of the spleen and the lymph nodes. This has a certain up and down pattern to it, but you note that the thymic indices remain low, showing a substantial atrophy of this organ, and this is associated with tremendous enlargement of the peripheral lymphoid tissues. These changes take place very early. The present interpretation of these results, without belaboring them in detail, is that the injected cells direct their attack primarily on the peripheral spleen and lymph node tissues. By feedback, the thymus is called upon to make good this damage, either with cells or hormone, and this results in atrophy of the thymus in conjunction with enlargement of these organs. By the time the animals are dying, there is a general lymphoid tissue atrophy, and this appears to be irreversible at this point. The animals go on to die. Now, lymphocyte populations have the capacity to protect against such transplantation disease as well. If isogenic adult lymphocytes are injected within 48 hours into animals that would otherwise go on and develop transplantation disease, such cells can protect the recipient from the killing effect of such lymphocytes from a foreign strain adult. Next slide, please. Now, recent views of lymphocyte involvement in immune responses in general have been summarized by a number of people during the last year or so. This summarization is from Turk and Stone, based on studies with contact allergens, dinitrofluorobenzene, dinitrochlorobenzene in guinea pigs. They observed that following application of the contact sensitizer to the ears that and use of tritiated thymidine label, that the earliest major response was the appearance of large pyroninophilic cells, to which Dr. Craddock has already referred. These appeared within about 4 days, and then subsequently labeled small lymphocytes in large numbers could be picked up in the regional nodes and elsewhere in the body. These small lymphocytes appeared about 5 days after stimulation, and plasma cells were not evident until 6 days at the earliest, not reach a peak till about 9 days. The interesting thing here is that a cytostatic agent, cyclophosphamide, was capable of blocking the apparent transformation of small lymphocytes into large pyroninophilic cells, and concurrently blocking the entire immune response following application of the sensitizer. When a folic acid antagonist, methotrexate, which is a very effective immunosuppressive drug, was applied, this drug was quite incapable of blocking the development of the pyroninophilic cells, but nevertheless interfered with the development of active immunity by blocking, apparently, the development of both small lymphocytes and of plasma cells. Now, the great mystery in this system is still the role of these large pyroninophilic cells. They're always present in the course of immune response. There's evidence based on isotopic labeling experiments that In fact, they transform into lymphocytes and may be derived from lymphocytes as well. Now, the shortcomings of this model are that one makes many inferences here about the developmental sequences involved from the behavior of isotopic labels, and this is not proved altogether satisfactory. Next slide, please. A more up-to-date summarization, perhaps, of this general evidence is as follows, that it's clear that the role of the thymus during early development is to control the appearance of small lymphocytes. If thymectomy is performed early in life, the most consistent general finding in all species that have been studied to date is a profound depletion of lymphocytes. So it appears clear that this developmental sequence from large to medium to small lymphocytes is controlled in a fundamental way by the thymus. There is still more recent evidence that there are at least two distinct subpopulations of small lymphocytes, one long-lived and one short-lived. Indeed, Amos Norman here at UCLA has recently presented evidence that a major population of small lymphocytes may well live well over a year. Now, these long-lived lymphocytes are very probably the cells that we and others have shown to be involved in transplantation immunity and in delayed hypersensitivity. When attempts have been made to adoptively transfer immunity with purified small lymphocytes from animals immunized to various antigens, infectious agents and soluble antigens as well, it has generally been found that the small lymphocytes are unable to transfer the capacity to make antibodies, serum antibodies, adoptively unless the hosts are re-stimulated with antigen. And other evidence strongly suggests that these small lymphocytes are immunologic memory cells. Another population, however, which is probably much more short-lived, is very probably involved in the serum antibody response via this pyraninophilic cell pathway, leading to the development of mature plasma cells, which, as has long been known, are the major cell type involved in immunoglobulin antibody production. This step here is a little more in doubt at the moment. It's clear from the thymus studies that although thymectomy early in life will profoundly impair this pathway, there seems to be a very irregular and indeed sometimes minimal effect on this pathway. Thymectomized newborns, although greatly lacking in small lymphocytes, are often capable of giving an excellent serum antibody response to a variety of antigens, and studies show that the levels of the major immunoglobulin classes are very little different from normal in thymectomized animals. Perhaps the most unequivocal evidence for a dichotomy in these two pathways comes from studies of birds, where in chickens it's established that the thymus controls the delayed hypersensitivity transplantation immunity pathway, whereas the bursa of Fabricius, a cloacal lymphoid organ, controls this pathway. Removal of the bursa blocks this sequence. Removal of the thymus blocks this sequence, and the two are more or less independent in birds. One could cite additional evidence, I think, for the essential separateness of these two pathways. It is nevertheless still unclear whether small lymphocytes are the only major class of cells involved here. They are probably not. It is, however, quite clear now that there are distinct subpopulations of lymphocytes that may be morphologically identical but have functionally very different endowments. Drs. Sparks and Skoog will subsequently consider with you the very exciting recent developments in the analysis of different immunoglobulin classes and their relationship to the cellular events. Well, in summary then, the thymectomized perinatal mammals consistently show subsequent depletion of peripheral small lymphs but have normal plasma cell and immunoglobulin levels. A specific humoral antibody response is only partially and irregularly impaired. There is a suggestion that the gamma A class of globulins, which is a minor class, is present in subnormal amounts in thymectomized animals, and just what the relationship of lymphoid cells to immunoglobulin classes may be is a matter that deserves and will no doubt receive much further study.
Dr. Sparks will go over briefly some of the evidence from in vitro culture studies indicating the potentiality of the lymphocyte as an immune cell. Dr. Sparks.
I wish to limit my comments to the human peripheral blood lymphocyte, although what I have to say about this cell may well apply to other lymphocytes, both in humans and other mammals. As been indicated already, the lymphocyte's function seems to be one of an immunological nature, and this will be the area that we'll wish to look at. Tissue culture studies of the peripheral blood lymphocyte are feasible because the lymphocyte, under techniques which are used today, is viable for a period up to 2 weeks. During this time, the function of the lymphocyte can be quite well studied. If one takes lymphocytes from the peripheral blood and looks at them, one will notice that they are primarily small lymphocytes. These are lymphocytes which have a relatively large nucleus and a small amount of cytoplasm. If you put them into tissue culture without any type of stimulation, they will essentially remain in this form and will not change. However, under appropriate stimulation in the same type of tissue culture, it is possible to stimulate these cells to transform and actually become functional cells in the production of antibody. Now, the cells on stimulation will transform into larger cells which will have considerably more cytoplasm than will the unstimulated cells. In addition, the DNA content of these cells will be replicated, and this can be demonstrated by autoradiographic studies with tritiated thymidine. The cells will also, under stimulation, will undergo cell division, and produce more cells. And in addition, they will be demonstrated to produce protein, which studies indicate probably are antibodies. It's been demonstrated that these proteins which are produced can be classified into the 3 classes of immunoglobulins using the present terminology, that is gamma G, gamma A, and gamma M. Fluorescent antibody studies also indicate that these cells are producing what appears to be gamma globulin. This is done by use of fluorescent anti-human gamma globulin, which will selectively attach to the cells which are producing gamma globulin. It's also possible by using precursors to the products which the cells are producing, such as radioactive labeled amino acids, and to show that the products which are produced will contain the amino acids, and on further analysis seem to be the gamma globulin or immune globulins. It's possible to stimulate the lymphocytes by specific and nonspecific means. Nonspecific stimulation is characterized or is typified by use of phytohemagglutinin. This is a substance which has shown considerable use in cytogenetic studies. By the use of this substance, it's possible to stimulate lymphocytes from an individual, and in so doing, being able to study their chromosomes. It's unknown how this substance acts. It's known to be a crude extract of the kidney bean, and it acts in a nonspecific fashion. However, in cells which have been previously sensitized, it will stimulate these cells to produce a specific antibody, the antibody to which the antibody will be reactant to the antigen which the cells have been sensitized to, as well as stimulate these cells to produce nonspecific gamma globulins. Now, in the specific type of stimulation, it seems that the cells will be stimulated to produce only the antibody against the cell, against the antigen to which the cell has been sensitized. In this situation, the cells seem to take longer to transform, and also a smaller proportion of the cells will be transformed than with the nonspecific phytohemagglutinin stimulation. These 2 factors, the time involved and also the proportion of cells which seem to be modified or stimulated by these substances, indicate at least that certain parts of these two stimulatory processes are probably different. It's also been found that by taking lymphocytes from one individual and mixing them with lymphocytes from another individual, that these cells will be stimulated, as indicated previously. That is, they'll go on to be transformed, go into cell division, and produce antibody. Although it's not clear what mechanism is working here, it's been postulated, or at least speculated, that this is probably an immunological reaction also. On this basis, it's been hoped that this type of test may be used to evaluate histocompatibility differences or immunological identity between donor and host in tissue transplantation studies. The in vitro studies have also been used to evaluate different types of clinical abnormalities, one of which is the sex-linked agamma-globulinemia, and some interesting results have been noted. It's found that these cells are cells from these individuals, lymphocytes, that these cells will be stimulated by phytohemagglutinin to be transformed and go into division, but they will not produce antibody with this type of stimulation. Now, with an antigen stimulation to which the individual has been exposed, the cells will not even be stimulated to divide or go into division and will not also produce antibody, suggesting that the two processes, again, may be somewhat different. That is, the processes of cell transformation and production of antibody. It's also been found that lymphocytes from an individual with Hashimoto's thyroiditis, a presumptive autoimmune disease, that these lymphocytes, when put into tissue culture and are exposed to thyroglobulin or phytohemagglutinin in tissue culture, will be able to produce gamma globulin, which seems on study to be an antibody to the thyroglobulin, suggesting that these cells are indeed able to produce antibodies to apparently normal tissues. I think in summary, the in vitro studies in general tend to support the concept that the lymphocyte is an immunologically competent cell capable of making antibody.
Thank you, Dr. Sparks. I might add that there is some evidence relevant to the patient that we presented that cells in culture from patients with chronic lymphocytic leukemia are incapable, or at least partially so, of the type of response that Dr. Sparks has mentioned in vitro. Another disorder of common encounter in which gross deviations in the immune system is characteristic is multiple myeloma. Here again, the dominant cell is a highly differentiated one, the plasma cell. Plasma cells in this disease are actively synthesizing globulin. The globulin produced in a given patient with this disorder is usually of a single molecular species, as though a group of cells, all with the capacity to produce only one type of immunoglobulin, had overgrown all other plasma cells, giving rise to the so-called monoclonal gammopathy, one clone of cells producing one gamma-type globulin. And this is characterized by a single sharp spike on the paper electrophoretic strip, reflecting the uniformity of molecular weight and charge of the myeloma protein. Plasma cell proliferation in multiple myeloma is neoplastic. It is not the result of any known antigenic stimulatory mechanism. The characteristic clinical features of multiple myeloma, for those of you who are not too familiar with the disorder, are widespread osteolytic bone lesions, dense accumulations of plasma cells in the marrow showing varying degrees of differentiation, anemia, hyperglobulinemia, and susceptibility to bacterial infections. Interestingly, the lymph nodes and spleen are not often involved early in the course of this disorder. Many of the features during the progression of a case of multiple myeloma are believed due to the changes arising from the overproduction of globulin. Amyloidosis, a common development in some varieties, is believed a deposition of a globulin polysaccharide complex in perivascular and connective tissue areas. Serious hepatic and renal derangement may follow, and in this instance, The amyloid deposition related to the hyperglobulinemia may lead to death as a result of organ involvement with a cell product rather than by direct invasion of the organ by the malignant tissues. Much has been learned about the structure of immunoglobulins, and a major contribution to this area has come from the study of these naturally occurring pure species of immunoglobulins in multiple myeloma. Dr. Sco will review some of these immunochemical developments.
It has been shown that plasma cells, as well as other cells in the lymphocytic series, are capable of immunoglobulin synthesis. It has long been known that the term gamma globulin encompassed a diverse group of protein molecules. With the advent of immunoelectrophoresis, three related yet distinct classes of gamma globulins have been described. Each class has been shown to possess antibody activity, hence the name immunoglobulins. May I have the first slide, please? This slide illustrates the three classes of immunoglobulins in relation to their electrophoretic mobility. As you can see, the gamma G class quantitatively is the largest, and immunoglobulins in this group may have electrophoretic mobilities anywhere from slow gamma to alpha-2 globulin by conventional paper electrophoretic techniques. These are 7S proteins with molecular weights of approximately 160,000. The gamma A class are immunoglobulins of similar molecular weight but have a larger carbohydrate content. The gamma M group comprise the high molecular weight immunoglobulins, usually with molecular weights of about 1 million. These proteins are also rich in carbohydrate. Next slide, please. Major non-lipid components of normal serum proteins are depicted here with electrophoretic mobility on one axis and the sedimentation rate by ultracentrifugation on the other axis. The immunoglobulins are shown as in blue. As labeled, most of the antiviral and antibacterial antibodies are in the gamma G group, while diphtheria and tetanus toxin antibodies and typhoid O and paratyphoid B antibodies are found in the gamma A group. The macroglobulin immunoglobulin group, or gamma M, include the isohemagglutinins and Rh factors, and pathological antibodies such as the heterophil antibodies, cold agglutinins, and rheumatoid factor are gamma M immunoglobulins. Next slide. Multiple myeloma, a malignant disease characterized by proliferation of abnormal plasma cells, is the classic example of a monoclonal gammopathy. A paraproteinemic hyperglobulinemia and or hyperglobulinuria is a characteristic finding in this disorder. This is a single abnormal globulin produced in excess by a clone of abnormal plasma cells and is seen as a tall, sharp spike on the analotrol tracing of a paper electrophoretic pattern. These paraproteins are most commonly identified with the gamma G immunoglobulin group. Normal immunoglobulins are suppressed in this disease as shown in this slide. This being the normal level, and this illustrates the suppression. The paraprotein may be identified less commonly with the gamma A immunoglobulin group. Another illness, but ordinarily a much more benign disease, is Waldenstrom's macroglobulinemia, in which a large molecular species paraprotein is produced in excess by abnormal lymphocytic cells. These proteins can be identified immunoelectrophoretically with the third immunoglobulin group, or gamma M. Next slide, please. This slide illustrates the current concept of the structure of an immunoglobulin molecule. As you can see, the molecule consists of two heavier H polypeptide chains, illustrated here in green, and two light or L polypeptide chains joined by interchange disulfide bonds, the L chains being in red, and the blue markers illustrating the interchain disulfide bonds. Next slide, please. Recent studies have demonstrated that the gamma globulin molecule may be degraded in various ways. Partial reduction by treatment with urea or propionic acid can separate the molecule into its component H chains with molecular weights of about 60,000 and L chains with molecular weights of about 20,000, as shown here. Mild reduction with dilute hydrochloric acid can split the molecule into two L and H chain components, while digestion with enzymes such as papain separates a portion of the joined H chains from the rest of the molecule. This portion has fast electrophoretic mobility and is called fast fragment or F fragment, while the remaining portions of the split molecule are called slow fragments or S fragments. S fragments being illustrated here in green. Next slide, please. This slide diagrammatically illustrates the polypeptide chains in normal immune globulins. The L or light chains of low molecular weight are common to all three immunoglobulin classes and are shown here in blue for each of the classes. These are either antigenic type 1 or type 2 and are found in normal serum in a ratio of 2 to 1. The heavier or H chains are different for each immunoglobulin class and are shown in a different fashion in this slide. Next slide, please. Pathologic immune globulins are depicted in this slide. Again, myeloma paraproteins can be identified with either the gamma G or gamma A immunoglobulin classes. Paraproteins found in Waldenstrom's macroglobulinemia are related to the gamma M class. It is of interest that the paraprotein molecules produced by the individual patient with these disorders contains either antigenic type 1 or type 2 L chains, but not both. A recently described paraprotein disorder, which has many features of a malignant lymphoma, is called Franklin's disease. Originally thought to be an illness in which abnormal lymphocytoid cells produced H or heavy chains in excess, it has been shown very recently that the paraprotein in excess is only a portion of the H or heavy chains and is probably identical to the F or fast fragment, which can be separated from gamma globulin molecules by papain digestion. Hence, fast fragment disease is a more accurate title for this disorder. May I have the lights, please? The peculiarities of the various monoclonal gammopathies are too numerous to be discussed further at this time. However, it should be emphasized that these disorders, in which a tremendous quantity of a single protein is produced by abnormal cells in the plasmacytic lymphocytic series, have been and will continue to be model sources for the study of immunoglobulin structure and function. Thank you.
Well, we're seeing a picture emerge of the specificity of immunoglobulins being determined by the amino acid sequence on the polypeptide H and L chains. In a manner analogous perhaps to the control of the amino acid sequence of hemoglobin alpha and beta chains, the sequences must be determined by its specific genetic loci. The genetic control of immunoglobulin synthesis is an infantile subject at the moment, but is destined to become another area of mushrooming knowledge. Dr. Sparks will give us a brief account of some approaches in this area.
Antibody molecules are proteins, and like other proteins, as Dr. Craddock has indicated, are basically under the control of genes. So we have the first slide. I'd like to just review briefly the structure of immunoglobulins. And you see here that this is very similar to the slide that Dr. Skog showed. It was taken from his. But the point I wish to make here is that the molecule as a whole is not just a single, made up of single chains. It's actually made up of 4 chains, 2 of which are H chains, 2 of which are L chains. But these are polypeptide chains, and polypeptide chains are made up of amino acids. We have the next slide, please. The primary structure of amino acids, or of polypeptide chains, is indicated such as here, where the amino acids are found in sequence. The polypeptide chain, the primary structure of this, is due to the sequence of these amino acids in the chain. Now, the relationship of the amino acids that are found in the polypeptide chain are, in a sense, collinear to the message which is contained up here in the DNA molecule or the gene. You will recall that in the gene there are nucleotide base pairs, and it's thought at this time that the sequence of these pairs taken in triplicate code for a specific amino acid, such as this group of base pairs might code for this type of RNA, which in turn carries the message down to the ribosome, so that, say, phenylalanine is coded for by this poly U here, and UGG might code for glycine, etc. So in this way, the structure contained in the DNA molecule in itself determines the primary structure of the polypeptide chain. Have the lights, please. Since antibodies are made up of polypeptide chains, one would expect them to be under genetic control, and studies to date support this contention. The simple structure, I think, of looking at the simple way of looking at the structure of the immunoglobulin molecule is that there are 2 different types of chains in each molecule. The 2 different types of chains, will occur in pairs. Now, there are the L chains, and these are under control of one gene. There are the H chains, and these are under control of another gene. Now, there are different types of H chains, and the different types of H chains are each under control of a different gene. In one molecule, there are again 2 H chains which are the same, and there are 2 L chains which are the same. I think for those of you who are familiar with the structure of hemoglobin, a brief look at the possible analogy that one can make between the hemoglobin molecule and the immunoglobulin molecule might prove valuable. We have the next slide, please. You'll recall that hemoglobin is made up of 4 globin chains as well as heme, but the structure that we're interested in are the globin chains. Now, in the 4 normal types of hemoglobin, hemoglobin A, hemoglobin A2, hemoglobin F, the structure is such that the 4 globin chains, there are 2 alpha chains and 2 beta chains, so it's alpha 2, beta 2. Going on down to A2, the alpha chains are A2 again, and here's a different type of chain substituted for the beta chain. This is delta chain. But in the F hemoglobin, the A chains are again the same, and there are 2 of them. But again, there's been another change, the gamma chain, and this again occurs as a pair. Now, this seems to be a fairly characteristic structure of proteins in nature, and the main consideration here is that the alpha chains are the same in all molecules, and it's the other chains, essentially the beta chains or its substitutes, vary in the different hemoglobin molecules. Now, to go to the immunoglobulin molecule, we have the gamma G, and these are some of the other terminologies which you may be more familiar with, but it should be pointed out that apparently there is considerable agreement now amongst those in the field that this terminology, gamma G, gamma A, gamma M, would be the ones most accepted. Now, the structure of gamma G is such that it has 2 L chains indicated here, and it has 2 H chains, which are gamma G H chains. You recall now it's the H chains which are going to vary in the different types of immunoglobulins. The L chains will be the same in all classes of the immunoglobulins, that is gamma G, gamma A, gamma M. The L chains will remain the same very much as the A chains do in the hemoglobin. And it's the H chains which vary, as you see here. So that in gamma A, the H chains, there'll be 2 of them, and they're the same. There'll be gamma A type, and the same holds true for the gamma M protein. the gamma M type of H chains are present. Now we have the lights, please. It's been found that the various chains, the H chains and L chains, since they are polypeptides or proteins, can act in an antigenic fashion. And on this basis, it's been found that these chains will react with certain specified antibodies in a certain way, such that the H chain will react with an antibody and is so designated as having an INV locus, or an INV antigenic makeup. The H chain has been similarly studied and reacts with an antibody such that it's designated as GM. As you probably know, there are different subgroups within the INV and the GM groups, and these can probably be accounted for on the basis of different alleles, that is, different genes, different alleles of the genes which go to form the different types of H chains, such that for the H gamma G, that is the gamma G H chains, that you can have one gene which will control formation of, say, GMA. You can have another gene which is allelic to this, which control the formation of another protein, which still is an H chain, a gamma G H chain, but it will be somewhat different, and because it is somewhat different, it may be designated GMB, so that the GM types can be looked on in this fashion as being under the control of the basic genetic makeup of the individual. Now, I think the next slide will help to bring all these things together. This is fairly complex, but I think as we go through it, it'll become a little more clear. And the way to look at it is, down here are the genes which control the polypeptide chains which go to make up the immunoglobulin molecule. Here we have the gene which controls formation of the L chain. Over here we have a gene which controls the formation of the H chain for the gamma G. Over here we have the gene which controls the H chain for the gamma A. And over here the gene which controls the H chain for the gamma M. Now again, each of these genes will produce its own product, its own polypeptide chain, as indicated here. Now we go up here to, again, the structure of the immune globulin molecule, and you recall that it is made up of 2 L chains and 2 H chains. Now the L chains are the same for the various types of immune globulins, and you can see the L chain from this gene goes here, the same L chain can go over here and over here. And these are the 3 classes of immunoglobulins. The gene which controls the formation of the H chain, gamma G H chain, will go into association, as you see here, to make a molecule containing 4 polypeptides, and this would be known as gamma G. Now the H chain over here, which is formed by the gene here, this is the H chain for the gamma A molecule. This will again go in association, so that there are 2 H chains which are the same, will associate with 2 L chains which are the same to form the gamma A type of immunoglobulin. It's indicated here that by N that this molecule may occur as a polymer, much as will the gamma M. Now the same story holds here, that you have a gene which controls the formation of the gamma M H chain. Here is the product formed, and it in turn goes up into association, as you see here. So in all the immune globulins, we have molecules which are made up of 4 polypeptide chains. In each of them, there are 2 L chains which are the same. The L chains are the same in all the molecules. The distinguishing features of each of the immunoglobulins is caused by the different types of H chains which are formed. Now, again, the INV, so-called INV locus, is located here on the L chain, and this will be found on the various types of immunoglobulins. The antigenic makeup of the gamma G H chain is such that it will determine the GM antigenic makeup, and it will be found only on the gamma G molecule. It will not be found on the gamma A or the gamma M. Now, I think, at least I hope this brings the picture together. It does for me, in trying to show the relationship of the genes to the formation of the immunoglobulin molecules. Now, it should be noted that on the basis of studies to date, that all these genes are found on autosomes, that is, the non-sex chromosomes. We don't know which autosomes they're located on, whether they are found linked to one another or not, but at least they're found on autosomes. Now we may have the lights, please. I think those of you who may be familiar with the sex-linked congenital agammaglobulinemia may ask, how does this condition relate to the problem that we see here, since it seems that the genes, the structural genes, which cause or determine the structure of the immunoglobulins are located on the autosomes. How can a gene located on a non-autosome, that is on a sex chromosome, have anything to do with the immunoglobulins? Well, it's found that in the agammaglobulinemia, the sex-linked type of agammaglobulinemia, that all the immunoglobulins are depressed. The current concept is that there is a gene located on the X chromosome, an X-linked or sex-linked gene, which in some way acts in a controlling fashion, possibly controlling the formation of the L chains, in a sense acting as a controlling or regulator gene, and in a sense determining whether these immunoglobulins will be formed or not. So I think this may again explain the relationship between the autosomal structural genes, which control the structure of the immunoglobulins, and a gene located on the X chromosome, which also can determine the amount of immunoglobulin present.
Well, you can't help but wonder how Dr. Sparks's charts will look when he takes into account all of the various antibodies and how they specifically arrive. Our anchor man today is Dr. Aquila, who has a special interest in immunology and is going to try to pull together some of the pathology and its relationship to some of the things that we've discussed today.
Gives me what, 4 minutes? When anyone tries to put together a mosaic picture of diseases, he always winds up making an arbitrary classification. So if you will permit this arbitrary classification, I would like to preface it with the idea that this is arbitrary, that all of these diseases probably overlap in some fashion. Can we have the first slide, please? I've more or less divided the diseases related to immune globulins into 4 general classes. Diseases wherein there is a deficiency of an immune globulin, some aberrant inability to synthesize either all of them or part of them. Secondly, there are diseases in which there is abnormal synthesis, a bizarre type of production, and these, interestingly enough, are diseases related mainly to neoplastic states. They produce neoplastic globulins, such as the myelomas, cryoglobulinias, heavy chain disease, and so forth. The other 2 diseases are somewhat related. There are diseases that are related to abnormal synthesis of intact antibodies, that is, antibodies which are capable of reacting with antigens. These may be possibly related to the ability of whether or not the patient is tolerant to certain tissues. That is, has he lost tolerance, and is there some sort of a release mechanism allowing synthesis against his own tissues? In the autoimmune diseases, I've remained and kept it strictly and arbitrarily diseases produced in relatively normal individuals, normal from the point of view of their ability to produce antibodies and their tolerance to their own tissues. These are diseases produced by organ-specific antigens. It should be emphasized at this point that these 3 disease states, interestingly enough, are somewhat related clinically. As persons with rheumatoid arthritis and lupus will often have familial people very close to them who will have agammaglobulinemias, and people with agammaglobulinemia frequently will develop rheumatoid arthritis. So that this relationship here I would like to point out to emphasize the fact that this is probably a continuum. It's a mosaic, a large disease pattern, and these subdivisions are arbitrary. I'll have the next slide. In the deficiency disease group, these are just examples of them. We have, of course, the 3 circulating humoral antibodies, which we can identify, we can measure chemically, we can tell you all about the chains. What we don't know much about is the cellular attached antibodies. And interestingly enough, the prognosis of these individuals does not seem to be related to the presence or absence of the globulins so much as it's related to the presence or absence or disarray of the cytological elements involved in these disease processes. Those infants where there is no thymus, where there's almost complete leukopenia, complete absence of plasma cells, regardless of substitution therapy, do very poorly. They die very young. Whereas in people that have the absence of all 3 immunoglobulins, which have intact lymphoid tissue, they reach adulthood, and some of them are still continuing on in fairly normal fashion. So there is a dichotomy here, reference to the clinical course that these things will take. I suspect that we're going to probably get a whole host of diseases. That's why I emphasize examples. And every day, if you pick up the journals, you find that there are different types of diseases. Here's a disease with an absence of the gamma A. It has normal gamma M, and either normal or depressed gamma G. These are infants with ataxia and telangiectasia, which have a dysplastic thymus and a leukopenia, specific disease entity related to a deficiency in this group of immunoglobulins with a relative leukopenia. There are various forms of these dysgammaglobulinemias, and I won't go into them in too much detail since time is short. May I have the next slide, please? I've gone over skipped over the diseases. Could you turn that off a minute and give me the lights? Relating to the proliferative states. Dr. Skog mentions the multiple myeloma. He mentioned the heavy chain disease and the macroglobulinemias. I would like to emphasize here again that there will probably appear a series of other diseases related to these aberrant proliferative states. Probably the next group will be heavy chain related to the gamma A and heavy chain disease related to gamma M. They haven't been defined yet. They probably will in some malignancy somewhere along the line, and I'm sure there are people looking for them at present. As a matter of fact, it's interesting to note the heavy chain disease, the gamma M heavy chain disease was predicted before it was discovered by the people that were interested in that particular area. Can I have the next slide, please? In those conditions where we have an abnormal synthesis of intact globulins, or we might term them as a disturbed tolerance, there's a series of things which I think we could say in general. They are predominantly female. These diseases overlap with one another. You find disease features of lupus and rheumatoid arthritis and Hashimoto's thyroiditis in varying degrees within the varying individuals with the diseases. They contain a multitude, frequently, of humoral autoantibodies, antibodies against nuclei, antibodies against red cells, antibodies against platelets and white cells, and antibodies against practically everything that you want to look for, I guess. The antibody level that the people manifest, such as in rheumatoid arthritis, the level of the 19S rheumatoid factor, doesn't necessarily relate to the pathology nor to the onset or cause and effect of the disease. There's, again, I'd like to emphasize the familial incidence in all of the immunoglobulin diseases. And then there is a familial incidence not only of the diseases, but there are many people that will have aberrant immunoglobulins, familial, related to people with these diseases that won't have the disease. So again, this is probably a mosaic of disease involving some genetic abnormality in this immune system. Now, add the last slide, please, or next to last slide. I would like to emphasize that in the autoimmune diseases, probably the important thing is the organ-specific antigens, and the hallmarks are the lung, brain, thyroid, and adrenal, and there are other tissues that are have been shown to contain organ-specific antigens. These are antigens that when injected, and they must be injected in Freund's adjuvant, will produce antibodies against these antigens and frequently induce pathologic changes either in the brain or thyroid. It's interesting that the antibodies that you can measure against these antigens, when passively administered to the animal, will not produce a disease. In order to produce the disease to the thyroid or to the brain, one must administer the lymphoid cells. And again, here we have a dichotomy between the disease process and the circulating humoral antibody. Here we have to use the cell, and it appears that there's some sort of a cellular relationship to the disease process. Probably the best example of a strict autoimmune disease is the allergic encephalomyelitis following the old Pasteur vaccination. And here, this is a demyelinating disease which occurred because of the organ-specific brain tissue or central nervous system tissue from the rabbit in which the rabies was grown. When injected into the people that were being immunized, they produced demyelinating diseases. I'd like to contrast this at one pole. That is a disease caused strictly by an organ-specific antigen. At one extreme, to the diseases in systemic lupus at the other extreme, where the individuals have probably lost their ability to discern their own tissues, and indeed some of the lymphocytes can make antibodies against DNA which is present in the same cell. And just to part with the idea that the humoral antibodies don't do anything, I have a scheme up here showing those diseases which I think the humoral antibodies are definitely implicated along with the complement system. Can I have the next slide? This is probably a scheme which can be altered, and I just put it together. There's fairly good evidence that with circulating antibodies and antigen combined with each other, they undergo a physical chemical alteration. The evidence for this is by the augmentations in fluorescence and depolarization of light. Furthermore, you can take the antibody and cook it a little bit or polymerize it and activate this complement system without any antigen. So there's probably good evidence that before the first component of complement is converted to this enzyme in the presence of calcium, that the antibody is in some way altered structurally. There's good evidence that if one persists in immunization in the region of antigen excess, that a glomerulonephritis can be produced. This is demonstrated by Dixon down in La Jolla in a very elegant set of experiments. He showed that in this glomerulonephritis, both the antigen and the antibody are deposited in the basement membrane of the glomerulus in a sort of an irreversible fashion almost. Secondarily, after the first component of complement gets activated to this enzyme, it can activate the fourth component of complement, which in turn acts on the second component of complement. There's an inhibitor in tremendous excess circulating in most individuals' circulation, which blocks this esterase activity. Recently, there's been described an hereditary disease where this inhibitor cannot be synthesized at all by a certain individual or synthesized to varying degrees, in which case, when the first component of complement is activated, it acts on these two components, and in these individuals who develop this angioneurotic edema, which can be life-threatening at times, there is also a marked depression in the fourth and in the second component of complement. So here you have an example have a humoral mechanism which is causing a specific disease entity because of a lack of the synthesis of this protein inhibitor. And finally, down the line here, and probably in some way unrelated, and might be other systems coming in here from the clotting mechanism and elsewhere, is a third component of complement system, which now has 4 or 5 different mechanisms, and it is involved in immunohemolysis, red cell immunohemolysis, and the Arthus reaction. And when it is acted upon, it also liberates leukocytic substances now, which attract polymorphs and set up inflammatory sites. I've tried in a very cursory fashion to give you a brief summary of how I think this immunological mechanism fits into certain disease areas.
Dr. Aquila, I think, has done a very fine job in a very difficult area of trying to coordinate what we've been talking about into actual pattern of disease. I think we'll have the remaining moments for questions from the floor. Would that be appropriate, Dr. Hewitt? Yes, sir.
I have a question for Dr. Sparks. In his system, where a gene is producing, well, a gene is basically responsible for the production of any antibody, does his system imply the pre-existence of genes for the production of all antibodies in response to all possible antigens that a body can ever come in contact with? Or is his system implying perhaps that new DNA or new genes can be formed during life when challenged by new antigen? Or am I going too far?
Well, I think the question basically revolves around the problem of selection versus instructive theory of immune globulin formation. And I think the system I presented could be compatible with either, and I should point out that actually the system I have illustrated here is somewhat simplified, that already there seems to be another class of immune globulins, gamma D, and there is also probably at least 2 to 3 different types of L chains. So it is possible that on the basis of the system I have illustrated here, that enough diversity amongst the antibodies which can be formed are already there in the individual as such. However, the concept of mutations, say, within the structural component, the structural genes, accounting for this type of thing could also occur. I would say, in general, the concept I put forth probably supports more the selective theory rather than the instructive type of theory.
It's certainly a very relevant question, and one that hopefully will have some more practical information about it, and not have to be so theoretical. Somehow the antigen can select the appropriate H and L chains, presumably, to make a specific antibody. Any other questions? If not, I think we'll... Yeah.
I just want to announce our program for next time, which I think is just 4 weeks from today. We're going to present about 6 cases to illustrate the newer developments in X-ray diagnosis. And these will be clinical cases, but with presentation of various features of new methods in Röntgen examination.